So I'll now, over the next 45 minutes, give you an overview of the challenges that we face. And as you see on the sign, National Dyslexic Association, or DNA, which I call a manuscript for a main actor without a role. So what is the scenario that we have today when we look at pediatric oncology in general? What you see here is survival curves from, in principle, the Nordic countries, but here also the Danish countries. Currently, when we did the last survey of the 5-year overall survival in Denmark, so that's every cancer from 1 to 18 years of age, the 5-year overall survival is 87%. But it's clearly an insufficient endpoint. So we want, because of the toxicity and still some patients that fail treatment, to have more individualized therapy. And we have the option to do that because all our patients are on protocols. They are quite deeply molecularly mapped, and we have quite good and very precise response monitoring. The good part is, of course, that all our cancers are in general very chemosensitive. However, with a very narrow therapeutic index, which means that every patient encounters severe side effects. We've got used to those like infections and others, but it's still quite severe. And for some patients, they will die from these toxicities. Overall, when we look at any cancer in patients above 1 year of age, at least in Denmark, cancer accounts for 20% of all childhood deaths. And if we look at leukemia specifically, half of all deaths within leukemia is caused by toxicity. So some years back, we established what we call the Ponte-de-Linea Toxicity Working Group. And I've listed on the left all the consortia or countries that participate in this activity. And what we did was we, in principle, step by step, go through a number of toxicities to provide deep phenotyping. I have on the right listed for which of these phenotypes that you all know, pancreatitis, osteonecrosis, thromboembolism, and so forth. Are there any publication that try to link these data to host genome variants? And that seems to be the case across the board. So it's not that we lack data. So why don't we use this more extensively and try to highlight you today on challenges that we face? So just to demonstrate that the Ponte-de-Linea group have been very successful, we've had a number of publications, several in the Lancet series, both on the toxicity definitions in general, on some specific items that we're now addressing. The first one was on pancreatitis. And more recently, we published a paper on what we call severe toxicity-free survival, which I think is a measure that you're going to account more deeply in years to come. And we have a number of ongoing research. We're capturing individual data on osteonecrosis in more than 1,000 patients, no toxicities in almost 2,000 patients, thromboembolism in more than 600 patients and growing, and so forth. So the way we normally define our outcome has been by overall survival or event-free survival, and we now want to add what we call severe long-term toxicities. It can be severe neurocognitive impairment, it can be organ failure of various kinds. So we all sometimes face patients that simply falls apart when we treat them. We've learned from the previous talk, for instance, by Madeline Youngman, where she highlighted that patients that have excessive toxicity, they may have an underlying DNA repair syndrome. And it's quite clear that some of these very severe toxicities may ultimately be explained once we start collecting data. It may ultimately be explained by underlying genetic variants. And I'll come back to how we can go into address that, but that's something that clearly will be a goal in the years to come. Another feature that is complicated when you want to understand how the genome affects a phenotype is that our patients are very diverse. I mean, if you study young adults or even adults in general with a cancer, they're much more homogeneous. But when we understand a phenotype on a specific organ toxicity, our organs don't grow proportionally. So we have this very, very rapid growth at birth, where we grow like almost 1 kilo a month. And of course, that will affect all organs because they're rapidly changing. If we look just at the proportion of the body at at birth, the center of the body is mid-abdomen, at the adult is between the legs and the body mass index and metabolic rate changes. At 1 year, 2 years of age, where a lot of our leukemias start to emerge, we have 20% of adult height, but we have 50%, 20% of adult weight, but we have 50% of adult height. At birth, our head is 25% of our height, but when we adults, it's only 14%. And the brain at 6 months is 50% of adult size. If you look at the plot on the left, you can see the rapid brain growth within the first years of life, and you can see the rapid growth of the thymus within the first year of life. And of course, this organ will be vulnerable for that specific reason. So when we study children with cancer, whether they are infants, youngsters, or adolescents, it's a different body composition that we try to address, and we don't really have measures on how we're going to adjust that. So this is some of the challenges. For leukemias, we some years back made a study where we compared young children, adolescents, and adults, because the leukemia protocol that I shared for the last 10 years, it encountered or encompassed every patient between 1 and 45 years of age. And what you see here is clearly that the adolescents and adults are very close in their toxicity profile, but very different from the younger children. So it's usually not something we take into account if we address a specific toxicity like pancreatitis or thrombosis. We just tend to lump patients together. So we need very large data cohorts to be able to compare patients across these age ranges and understand is a specific genomic playing a role in a specific age group. I'll come back to that issue with a few examples. So what is the cancer patient trajectory that all our patients face? So first, of course, it starts off with tumor biology. And of course, the tumor genome plays a key role there. Then you have some frontline therapy, and it's very often adapted to the tumor genome. Certain aberration will require certain therapy, like with Philadelphia, with 1221, higher hyperdiploid, and so forth. Sometimes treatment fail, and again, you use tumor genome sequencing to address phase 1 and phase 2 therapy. But that's only one of the genomes that are in play. The other is, of course, the background genome or the host genome. It plays a role for etiology. It plays a role for pharmacology. It plays a role for our acute toxicities. And of course, it plays a role for the long-term toxicity, and that is late effect on how we re-epithelialize the patients. And of course, in combination, the tumor and the host genome, they interact to preserve cure. So addressing the DNA is in general of significance, but we need to address them both in order to improve our therapy. But if we map the genome, and here on the x-axis, I have the allele frequencies going from very, very rare, including the private mutations that each of us harbor, to the very common ones. And on the y-axis, I have the effect size. So how should we understand that within pediatric oncology? So if we take the cancer-prone syndrome that we have addressed today, you could say these are very rare variants, but they have a huge impact. If you take the common variants, it's very rare that they have a huge impact. The two best examples is the Leiden factor 5 mutation that predisposes to thrombosis, at least in normal individuals. And of course, there are cases with abnormal pharmacology like the TPMT genotype. The problem is that most of our patients, they are within a range where we have relatively low frequency or common variants where the impact on our phenotype is very limited. As you see on the effect size y-axis, the effect is in general only modest. So we may need polygenic risk scores to integrate all that. Let me just looking at this slide composition. Let's take one example, leukemia etiology. So this is from a textbook that chapter that we published a few years back together with Jun Yang. So on the lower right, you have all the SNPs, and there's a growing list of SNPs that seem to influence either leukemia risk, ALL risk in general, or only some subsets. And then across the line with lower and lower frequency and higher impact, you have all our syndromes. You recognize Li-Fraumeni syndrome, ataxia telangiectasia, constitutional mismatch fat deficiency, Fanconi anemia, Down syndrome, Recklinghausen, or Robertsonian translocation involving chromosome 21, where you have a massively increased risk of interchromosomal amplification 21. So now there's a growing list also of ALL syndromes. Some are common. The ETV6 is probably as common as the Li-Fraumeni, but you have a number of other rare variants that may cause an increased risk of leukemia. But does it play a role in our daily life, and how frequently do we encounter these patients, each of us? That's less well mapped. So with this data and our ability actually to map the full genome of any of our patients, why haven't pharmacogenomic-based personalized medicine been implemented? Why has it failed so far? And I'll highlight some of the challenges and then give you some examples on where we do have some data. So genotypes are very precise. We can on every side of our 3 billion genome in an individual patient say exactly which of the 4 nucleotides are located there. So they're very reliable, and we often sequence our genome like down to 30x or sometimes even 100x. So we know what we have. But phenotypes are very poorly mapped, which means that you can take a patient that have thrombosis, but the risk of thrombosis will vary between our patients because some will have a central line, others will not. Some will be very mobile, others will not. And they have a different genetic profile. So the phenotypes may map differently. And human biology is in general very complex. So, and it's redundant. So even if you have a gene that in genomic association studies have shown to be important, when we knock out that gene in an animal model, sometimes nothing happens because our system is redundant and others will take over. So we have very few variants that have a recurrent, highly predictable impact. And the other option is, of course, that we have phenotypes that we use for prediction. If I have a patient that had very, very severe toxicity with massive infections, I will be less prone to treat this patient intensively. We use the example of maintenance therapy quite frequently because there we clearly adapt our treatment intensity to another phenotype, that is the blood count. Or if we want to do prophylaxis for thrombosis, well, we pick the teenagers and adults. So we have some phenotype issues that we use to adapt our therapy with anyway. And then if we want to combine multiple variants to have a polygenic risk scores, we really need very, very large data sets. And most pediatric cancer studies only include often less than 500 patients, sometimes even less than 100 patients, and very rarely do they go to 1,000. And that takes away the necessary power to do good genotype-phenotype studies. So if some of the clinicians feel, why are we so bad at doing personalized medicine based on the genome? Well, we really have tried. We need to have a different way of addressing it. And as I mentioned before, we now have quite good global collaboration, at least to do the phenotyping better. And then lastly, statistical significance is never the same as clinical significance. And even findings published in very high ranking journals are rarely clinical relevance. So let me just give you an example. So let's imagine we wanted to do a research on osteonecrosis. We had 1,000 patients and 60 of those developed osteonecrosis. And then we defined 2 pharmacogenomic groups, a group A that constitute 90% of the patients. And among those, 5% turned out to develop osteonecrosis. We have a group B where 15% develop osteonecrosis. So the relatively risk is 3, and that can easily lead to a publication, sometimes even in Nature Genetics. However, even though the relevant risk is threefold increased in group B, still 75% of all our necrosis occurs in the group A. So even if we were able to eliminate by some intervention osteonecrosis completely in the group B, the overall occurrence of osteonecrosis will only be reduced by 25%. And if we did that intervention in group B, well, anyway, 85% will not gain from the intervention, which in itself sometimes can be quite burdensome. What if the scenario became even better? Let's say we have a polygenic risk score. We could now define the group B as a group that had a 33% risk of osteonecrosis. Well, still, even if we eliminated all of the osteonecrosis cases in that group B, we still were left with almost half of all the osteonecrosis cases, 45%. And again, 45% of our patients will not gain from the intervention. And that is probably why clinicians have been so conservative about preemptive interventions, even though a lot of publications have come out, and we never see hazard ratios or relative risk of 11. They're typically in the order of 2 to 5%, 2 to 5. So when you read pharmacogenetic paper, you'll get a lot of data on the GVASs of minor allele frequency, of relatively risk, and beautiful p-values. What you need to explore in the papers, and very rarely, it's never written in the paper, is the proportion of patients accounted for by the genetic score. That's really the critical information, because that is what we're going to use for personalized therapy. So what should be our targets when we want to address the host genome beyond the syndromes we've talked about previously today? So we can gain a lot from these genomic profiling. We can get a deep understanding of biology or pharmacology that underlines the phenotypes, and sometimes that can lead to changes in our strategies, even though it doesn't necessarily allow us to do genomic-based personalized medicine. Sometimes we realize that the variance modifies the CPS phenotypes. There was a recent great paper by Christian Kratz on TP53 variants, several thousand patients with TP53 variants, where it was quite clear that the mutations in the critical mutation in the gene itself did not always lead to the same Li-Fraumeni syndrome. So the phenotype may change. Also, there's papers that identify genetic modifiers to take the Li-Fraumeni patients. Again, the MDM2 SNP variants clearly accelerate the tumor development. So it's not a syndrome itself, but it's a variant in the rest of the genome that influence how the cancer-prone syndrome manifests itself. Some of the very great stuff, and I'll come back to that in a moment, is on drug dosing, CPMT, NOT-15, CEP72. But even though the data is quite convincing, it's not universally implemented in the clinical care, even though doing the genotyping is much, much less costly than treating the patient just for one day. We can do polygenic risk scores, that is adding multiple small but additive effects, but again, we rarely have the study sizes that allows us to do it. Sometimes the genotyping can be used for defining high-risk populations. So let's say you want to prevent thrombosis in leukemia therapy with asparaginase. Instead of giving low molecular weight heparin to every patient, you may say, well, if a genotype can target a specific subpopulation, I could do my testing in that population and get higher study power. So once you've accepted the intervention as something that is acceptable for the patients, then you can use the genomics to limit that population. That's a case where it can be beneficial. And then lastly, every time we do our rounds, we adapt our therapy in some way. We decide whether the patient can start therapy or not, whether you should give more antibiotics or not, whether you should re-expose the patient after toxicity like pancreatitis. And that is very individual. I may want to do something with my patient where Jelena will do something differently. Having a genomic profiling may sometimes help you in that decision taking. So we take the decisions anyway in treatment adaptation. Even a modest guidance by genomics would be something that clinicians may accept rather than changing the therapy completely. So these are some of the, you could say, lower hanging fruits where genomic profiling at least can be beneficial. So for those of you that don't work a lot with genomic in the daily life, I'll just give you a few examples on how I see it. So on average, our nucleotide sequence differ something like 1 in 300 in a Caucasian population. If you look globally, 1 in 50 of our nucleotides will vary across the globe. So it's quite frequent variations. And only, as you know, 2% of our genome codes for proteins. What is this size really? When we're talking about something that is approximately 1,000 times Shakespeare's complete works. He wrote 43 works, and that's approximately 4 million letters. So approximately 1,000 more letters we have in our genome. So it's quite massive, and we really don't understand it at all. We are with cancer-prone syndromes and some of these variants just picking the very, very low-hanging fruits. We're going to see a massive development over the next 50 to 100 years. The most common variants are the single nucleotide polymorphisms. So these account for like 90% of the genome, and they're very randomly defined as a minor allele frequency above 1%. Please note that once we define the minor allele, it gets stuck, even though the minor allele in some population may be above 50%. So each of us, as individuals, we may have 10 million variants. I've just taken 2 twins on the side. Beyond those, mirrors of each other's, the rest of us is really, really different. The good thing is that DNA is inherited in blocks. So we talk about haplotypes, approximately 5 to 20,000 base pairs, but you can also talk about topological associated domains, which are larger constructs of the genome where various regions interact with each other. The good thing about the haplotypes is that they allow us to do imputing. So what is imputing? Well, before I come to that, I'll just say a few things about genotype-phenotype association studies. So when we do these association studies, we sometimes take a candidate gene association approach. It can be we validate some other group's genetic findings. We have some biological underlying knowledge. Let's say we know the genes that codes for coagulation factors, so that can be used. Sometimes we know the mutations in the tumor, well, was there mutations in the same gene in the germline? So we have this crosstalk. And as I mentioned before about the leukemia, ETV6 mutations are seen very commonly in leukemia, now also in the germline. The ICROS mutations are seen very common in leukemia, now also in the germline. So we know that there is this interaction. We can do conventional genome-wide association studies, and I'll give you a few examples for that. Just mention, why do we have this cutoff p-value of 5 times 10 to minus 8, and it's almost become religious, which is really nonsense, but that's the case. Well, it's simply to say if we study 1 million variants, instead of just one association, well, you have to multiply your normal set limit 0.05. You have to multiply that with 1 million. That's how you come to the 5 times 10 to the minus 8. You can do pathway association analysis. You can do polygenic risk scores, and you can do machine learning. But deep phenotyping, as I said before, is a must. And if you want to do these studies, you need very, very large data sets for polygenic risk scores and machine learning. And very few have really accomplished that yet. So a few things about imputing. So normally what we do is we take a SNP profiling platform. We genotype on that platform, and it's relatively cheap compared to whole genome sequencing. And then you see whether any of the variants that reacted genotype was associated with a specific phenotype. So that's the plot B on the lower left. Now, because we inherit our genome in haplotypes, what you can do is that you take all your SNPs on your SNP profiling platform, and then you phase it, so you localize each of the variants to your chromosome region by your reference material. And then you take a reference set of haplotypes where we have mapped every single variant in a specific region. So it can be HapMap, but it can be other stuff. And now more frequently it becomes use the whole genome. Now, the nice thing is because we inherit our genome in haplotypes, it means that if I within a haplotype, so you could say that if I within the red bar here, listed here, know most of the variants, I can impute or guess the rest. So 2 patients may have the same variant in one size, but they have different variants on the other. So they have different haplotypes, but in the end, they may have overlapping genotypes in the regions that we didn't put on our SNP profiling platform. And this means that suddenly you can markedly increase the power in the genotype-phenotype studies. And you can reach a GWAS significance. So that's something that's done routinely when you do GWAS analysis. You don't just take the, let's say, 300,000 or 500,000 SNPs that you have on your platform, but you can expand that up to perhaps 5 to 10 million. And that may provide much more benefit. It's not always successful, but it's quite frequently successful. So let's take some examples on things that we have done in recent years. So first, a candidate gene project. So in the Nordic 92 and 2000 protocol, we used a lot of high-dose methoxate. So we went back and looked, and this was done in Denmark. I'm living in a small country, just 5 million people. So all of you who are living in larger population, you could have done this work much, much better. But we could take this Danish population, treat it on true protocols. And what we knew when we use high-dose methoxate is that in order for the drug to work, it needs to be carried inside the cell, in this case, the leukemic cells. And for that, the cell used the reduced folate carrier. And there are quite common polymorphisms in the reduced folate carrier gene. So we mapped the most common one in the reduced folate carrier, and we took the variant A, T, A, and GLLs. And they're coded on chromosome 21. And what we found was that the patient that had the AA variant, which would be more profoundly transporting the methoxate into the cell, they had half as many relapses, so an event-free survival of 89%, compared that only had either one allele or no A alleles. These patients had an event-free survival of 80%. It was even most pronounced for the T cells, but that's another issue. But what we did then was we said, well, it's coded on chromosome 21, and we know that approximately half of all our leukemia patients, they have an extra chromosome 21 in their leukemic clone. So we looked at the interaction between the leukemia genotype on chromosome 21 and the outcome. So for patients who had 2 chromosome 21 in their leukemic clone, the event-free survival was significantly influenced by the reduced folate carrier variant. But for those patients that had, you could say, a gene doses effect because they had 3 chromosome 21 or they had 4 copies of chromosome 21, the event-free survival was exactly the same. So this is just an example of a candidate approach, but more importantly, that there was an interaction here with the germline variant and the tumor mutations. Another group, this is from St. Jude, they took a genome-wide association approach. So they took a number of patients. In the first study, they had 434, and in the subsequent study, they had 699 deeply sequenced patients. And what they found in their genome-wide association study, that this soluble ligand carrier, O1B1, was significantly associated with the clearance of high-dose methoxate. And when they did the deep sequencing, so they added not just the common variant, but also the private mutations, this gene accounted for almost 11% of the variation in methoxate clearance. This is nice from a, you could say, scientific point of view, but it doesn't help you much because you could say 90% is still not accounted for, but it's a demonstration of how much you can get from the GWAS study. Let's look at thiopurines, one of my favorites. We have 2 genes that influence thiopurine metabolism, the TPMT gene and the methyl thioinosine monophosphate even inhibits normal purine de novo synthesis, so that can enhance the incorporation into the DNA. Another variant, not T15, breaks down the triphosphorylated nucleotides, so you have less incorporation. And Jun Yang from St. Jude did a study back in 2015 where it demonstrated not just that both TPMT and not T15 variants played a huge role for tolerance to thiopurines, but also that the frequency of the variants varied across variance groups. So Africans have quite commonly TPMT variants, but hardly ever not T15, whereas East Asians very commonly have not T15 variant, but rarely TPMT. So you could say we may have the same phenotype, but that's explained by different variants in the genome, host genome. We looked at DNA-TG in thousands of samples of patients that had a TPMT genotyping. And what you see is that those that had the TPMT low activity variant, they had higher DNA-TGN during maintenance therapy. But you see on the right, on the black and the red curve, that even though it's slightly higher and get higher and higher during therapy, there's a huge overlap. And if we look at the risk of relapse, the risk of relapse was almost the same for the TPMT heterozygous and wild type patient, 6.4 versus 6.0%. So it doesn't really help us in our therapy. We did then a GWAS where we tried to understand the incorporation into the DNA. And what we found was when we did that on more than 400 patients and more than 4,000 blood samples, we did find a very significant finding on the explanation on the propensity of incorporation of thiagonucleotides into the DNA. And the interesting is that this top SNP, which was highly significant, was in a gene called NT5C2. And some of you may recognize that gene because it's been very frequently found in patients with leukemia relapses on therapy. So we added to that a study where we tried to link NT5C2 variants in the leukemic relapse clone with the germline variants. And what we found was that the likelihood of harboring this variant in your relapse clone was significantly higher if you had the germline variant. So you had the GALL compared to the GGLL. So what we anticipate is that on one of your alleles, you already harbor the germline. If you then in addition have an acquired mutation in your other allele, then the leukemic cell become resistant and that can lead to relapse. I'll skip this one. Then we take a candidate gene approach and take a common toxicity like thromboembolism. So this is another approach you can take. You have a phenotype of interest, in this case, thrombosis, and we look at which variants have been found in adult studies. So this study encompassed almost 60,000 individuals that were either cases, 7,500, and controls. And we took the 4 top variants, factor 5, factor 11, fibrinogen, and ABO blood types, and we looked at those in a Nordic study. So we had these 4 top SNPs that was found in adult thromboembolism, and then we looked, were any of these single variants or in a multiple regression analysis associated with a risk of thrombosis. And the 2 that came out significant was fibrinogen and factor 11. But note, not factor 5. So Leiden factor 5 mutations may play a role in general, but that role is not enhanced in our leukemia patients. But then to that, we added the age of the patient. So what you see here is the cumulative hazard plot for patients that have, and it should be 1 to 2, not 0 to 1, 1 to 2 alleles or 3 or 4 alleles. And what we found was that in the younger children, the risk of thrombosis were low, whether or not you had the genomic profile. In the adult patient 18 to 45, the risk was high in all cases. But for the adolescents 10 to less than 18 years of age, the risk was almost 4 times higher if you had this allele risk, so 3 to 4 alleles, compared to you have less than that. So you could say if we want to study thromboprophylaxis, the adolescents may be the group that we could use the genomic profiling. So you could say, okay, we want to include all adults, and then the adolescents, but only those that have this allele profile. Another variant that people have looked at is the variant that causes neuropathy. When you give vincristine, this came out from a large DVA study, again from St. Jude and COG, where they found that those that have the TT alleles or genotype in the CEP72, this is a gene that plays a role for producing microtubules, and you know vincristine interacts with that, so it's not a surprising finding. And it could also be validated in cell lines and stem cell neurons, but at least it was very profound and validated both in pediatric and adult studies. And now St. Jude is running a randomization where patients with the variant is giving either a lower or shorter vincristine dosage. So this is a testing on whether you can actually implement this finding in real life. And then I want to move on in my end of my talk to talk about more complex polygenic risk scores. So this is a study we initiated some years back. So we say normally the human body have, because we have these 20,000 genes, we have probably at least 10,000 proteins, and we have 500,000 protein-protein interactions that are known today. So proteins bind to each other and modulate the effect of each other. There's multiple databases, MIN, BIND, DIP, GRIDS, Reactome, and so forth, because not all bioinformatic researchers agree on the issues, but at least we have quite good knowledge on which protein interact with which proteins. So what we did is to say, what if we take a genome-wide candidate gene approach? So we went through all the literature that has been published on any kind of protein-protein interactions in pathways that play a role for leukemia. So we took the targets or the genes involved in all our 13 routinely used anti-cancer agents. We took targets on a number of toxicities like inflammation, coagulation, bone metabolism, DNA repair, and so forth. And we took all the first order protein-protein interactions. And then we said, what are the variants that we know about in these genes? And in the first round, we came up with 25,000 variants. In the next one, we came out to 35,000 SNPs, which covered approximately 74% of our reactome pathways. Then we made a platform, we used the adjuvant SureSelect, where you could extract specifically these 35,000 SNPs. We then barcoded them, we pooled them across patients, and then we sequenced them all. And then we could cut the price down for this very extensive sequencing, which was quite low, to €300 per sample 10 years ago. So that was really minimal. And then we started to doing our polygenic risk score. So the principle was that we took not just single SNPs, but we took SNPs that worked within the same pathway. We generated combination of 3 SNPs of whatever combination in these pathways. And then we used neural network to build prediction models for the risk of relapse. So we started off with 3 SNPs, and then we added 1 by 1 additional SNPs up to a maximum of 15. And then we took the 200 best combination to see how can we predict the risk of relapse. And then we looked at what are the pathways. So remember, now I moved from candidates and GWAS to pathway analysis. So we don't say, what are the most important variant that cause the phenotype, but what is the most important pathway that cause a phenotype? And the top pathways that came out was steroid metabolism, which is nice because we know that steroid is the most important drug in anti-leukemia therapy. Then we found also that pathways involving doxorubicin and methotrexate played quite a critical role. And then we subsequently did a regression tree analysis. So we took, you could say, tree branching. So what was the top categorizer of patients for relapse? The top was MRD. The next one was white cell count at diagnosis, but only for those that have a high minimal residual disease at end of induction. And then came a number of pathways involved in steroids, but also others like folate metabolism, purine metabolism. And based on this, we could distribute the patient that we addressed, it was 2 Danish cohorts and a German validation cohort. So in total, almost 700 patients. And we could now divide these patients into 3 cohorts. A group that was almost 80% of patients, 77.2, that only encountered for 3% of all relapses, and a group that only encountered for 7.5% of all patients, but 72% of all relapses. So seemingly a very powerful tool for relapse prediction. So this is just to emphasize that sometimes you can get very far even with a relatively limited number of patients, but we were close to 1,000. But the bioinformatics that you need is then extremely complex in machine learning algorithms and neural networks. And then in the end, I want to just say a few words about the ELEGANT study mentioned before. So it's an acronym of Exploring Leukemia, Education, Genetics, and Novel Technologies. And it's a sub-study under all the 3 European consortia altogether, IFBFM and ALEC. And ALEC also includes Australia, also includes Chile, Uruguay, and Argentina. And IFBFM also includes Australia. And we want to use this for developing, deploying, and utilizing very large data sets in this unprecedented collaboration. So these are the countries included, 33 countries, and we want to compile data for at least 15,000 and hopefully 20,000 patients over the coming years. And all the organizational process has been initiated. So we're getting informed consent forms across and ethical approval across these countries. And hopefully the samples collection from the first countries will start here in June. It's a complex acronym for collecting data, but the basic principle is that we get the blood sample, we do SNP profiling, and then we store it in a data repository. And then researchers that have a question on any phenotype, it may be treatment response, CNS leukemia, toxicity, relapses, MRD, you name it, can then apply for once they have scrutinized the phenotype in question across thousands of patients, then can then apply to link these data to the SNP profiles. The data is stored in Computerome, the life science platform in Denmark, which is quite powerful. We have more than 1,500 bioinformatic tools, so you can work with all your data inside, with all your software inside a secure environment and do your programming. And researchers and bioinformaticians sitting in Serbia or in US, or not in the US, South America, because they participate, or in Hungary, or in Italy, or in Denmark, they can collaborate within this network addressing the questions that they have. So what are the challenges we face in the years to come? The cartoon is, you need to do deep phenotyping, and I found a corner piece because we are really only at the start. So that's the most significant parameter, but it requires that you reach consensus definitions on that parameter. And just to highlight how complicated it really is, we just recently, in a large international group, published relapse definitions. So most of you would think that we know what relapses is in leukemia. Well, at least now it is defined, published in Blood this year. You need sufficient power to detect association, which means clinically relevant association, not just statistically. You need to have relevant odds ratios. Sometimes you need to register other parameters. I call it the exposome. So all the things that are around us, age, pubertal stage, and various exposures, chemotherapy, supportive care, microbiome, and so forth. You may add that, but it's not required. And then, of course, you need to map your omics. SNP profiling is one aspect, but other omics can be important as well. You need to do the complex bioinformatics. You need to demonstrate that any association you find can be validated by other groups, and hopefully also with a biological validation. And then, of course, you need to have a clinical applicable intervention. I mean, we can predict pancreatitis today, but we don't know how to prevent it. Or we can predict osteoporosis, and we're only starting to grasp how we can prevent it. But for thrombosis, at least we can intervene. And then, of course, you need to demonstrate that treatment adjustments by these host omics are better than adjustments by the clinical data that all of us use in our daily life. So this is the end of my talk, and I'm happy to take whatever questions you may have. So I stop sharing.
Yes. Thank you very much, Kjeld.
I would like
Yes, please.
to ask. Yes, Jürgen Ivanis from Austria. So thank you for this very nice presentation. It was going through the, I think, at the moment, future treatment options and analysis what you have. One of the questions is, if I would summarize your presentation, is my impression right that standardized treatment should move more and more to individual personalized medicine? Is this one of your key messages?
Yes, I think that's clearly needed. So that's most important. But we do it already. I mean, we individualize by age, by white cell counted diagnosis. We individualize by MRD or necrosis in a tumor after pre-operative therapy. So treating patients differently, we've always done that. The problem with genomics is that we currently adapt by phenotype, and phenotype is by definition a combination of very complex gene interactions. And now we try to boil that down by identifying single variants that plays a role, and it's simply impossible. You can only do this by very complex modeling, and that leads huge data sets, and that's why I engage in the elegant study. So personalized medicines will come, but it needs the complex bioinformatics.
Elegant, this project, what you are running, more or less a prototype project, we will build the future platform to run personalized medicine.
Yes. So one of the things that hasn't been solved that we're also addressing in part of the elegant is the legal requirements. GDPR is really limiting our ability to exchange very wide data set. And that's something we have to solve in parallel, but that's an activity that's ongoing.
If I would, the project on the one hand is from the generic point of view, we have all these, let me say, findings and models, the labs, we can, the genomic analysis too. And is it more at the moment a matter of artificial intelligence to get it run?
Well, that's one critical part. And bioinformaticians and biostatisticians need to crosstalk, which they don't do today. So that's one issue. The other issue is you need the data set. So we may, and we'll know that in a few years, be able to do this for leukemia because they're relatively common. But if you have liver tumors, it's almost impossible to do it unless you involve China and India and Nigeria, because you need so enormous amount of patients. But I think that's the way. And it may be that leukemia will provide information that can then be used for other cancers because we use the anti-cancer agents across different tumor types. That's the hope. But now we start with leukemia. And
Elegant, the project, is something what is running coming out from Denmark, or is this something which is supported by a global organization?
No, it's supported very strongly by the Altogether Consortium, by IOBFM, and by ALEC. But the data warehouse, the genomic data warehouse, and where you could say the bioinformatics will be run, is not by me. That's by all of you that have specific ideas. You will be able to, through your national consortium, to go through the approval process, and then you can address these specific issues.
But Kjeld, maybe you can mention there is 33 or 34 countries with
33 countries are involved, yes.
Great. When you start?
Well, the process has started. I anticipate the first samples will be collected in June prospectively, but many groups already have retrospective DNA stored that they want to include as well. So we anticipate to have approximately 3,000 new patients every year.
3,000 per year?
Yes.
Okay. Thank you. Great presentation. Thanks a lot.
Any other questions?
Kjeld, may I add some question here? Don't you think that to have just one big warehouse in Denmark is risky? I mentioned this actually. That
I saw you mentioned it, and I'm sure. 1 second. Schmiegelow?
Yeah, now we can talk.
Yes, I saw that you mentioned it. It's an issue we're going to discuss as we move forward. Federated data on genomics have been tried and it hasn't worked out. So if at one time point we get the option for be able to do genotype, phenotype data on federated data, I'll be fully supportive. This is the solution for the time being. We need clever people to solve that issue and it hasn't been solved yet.
No, I fully agree. But if there is another option, maybe to have a mirror site, because I mean, I'm not negative about what's going on in the world, but to have just one place where a lot of data are stored is very sensitive.
I can only say yes. Next question.